All right, everybody. Thanks so much for coming out to Boston Protein Design and Modeling Club tonight. Excited to see, I think, our largest in-person crowd ever. So yeah, fantastic to see the attendance. Just a couple of quick housekeeping things before we begin. So starting in January, we joined the OMSF, the Open Molecular Software Foundation. Really fantastic nonprofit organization that helps to support communities like ours. So we're really thrilled to be part of that. And before joining OMSF, we were unofficial, kind of informal collection of nerds that would get together and talk about protein science. Also means that we didn't have access to like a bank account or anything official to accept donations and really organize and have money. And we had folks come in and just sponsor our events by just catering. But as you can see by the turnout tonight, we're kind of outgrowing that model. So now we can officially accept donations. We're going to send out information soon. And part of that will also be sponsorships. So I know we've got lots of folks from tech and pharma and investment communities in the audience tonight. So we're going to be sending around things, asking for donations and helping to support this group, and also for all the the great food and drinks, and then also some swag. And so we'll have some really cool Protein Design and Modeling Club swag, t-shirts and stuff that will be supported through these donations. And that will also have the sponsor information on it as well. So the sponsors can be tied directly to the community. So that information is coming on the email listserv. So I think some folks, we have a pretty good word of mouth network as well. So if you come tonight and you've heard about it from someone else, you're not on our email listserv, please join. So that's where we send out all the information, where you can find the Zoom links. To join, it's very simple. Check out our website. We've got an email address. It's bpdmc@omsf.io. And that link's on our website. Just shoot an email, introduce yourself, say who you are, and what you do. Everyone's accepted. We just want to make sure that we don't allow bots to get onto the website and so onto the listserv. So if you just send an email, saying who you are and like a real human, we'll make sure you're added. Okay. Enough housekeeping. I'm really excited to introduce our speaker tonight, Sergey Ovchinnikov. So I think Sergey is one that pretty much everyone in the room knows, but I really enjoy embarrassing Sergey, so I'm excited to introduce him. So after an incredibly successful PhD with David Baker at the University of Washington in Seattle, Sergey moved out to Boston in 2017, I believe, and really has been instrumental in co-organizing this community since. Sergey was such a rock star as a graduate student that he basically got to skip the postdoc, and he moved out to Harvard as a distinguished John Harvard Fellow, which in Sergey's words is a pseudo PI. So not a tenure track, but still PI with all the downsides with fewer of the perks maybe. But was incredibly successful as a John Harvard Fellow. And as of January 2024, is now an official tenure track faculty member here at MIT, where we're having the lecture today. So super excited that Sergey is now official. And Professor Ovchinnikov, the floor is yours.
All right. Well, thank you so much. It's nice. Actually, I don't know if I'm going to use the mic, but if people can't hear in the back, feel free to tell me to use the mic and I could switch over. But I'll just stop it for now. Yeah. Thanks you guys so much for joining. There's still one seat here in front if anybody's still looking for a spot to sit. Oh, and also a few seats. There's a few seats in front here. I think there's only one person not sitting. But you have choices. You have choices here. Welcome you guys. And during the talk, feel free to interrupt me if you have any questions. I'm always happy to explain things a bit better. One other thing Chris didn't mention, but we're always looking for speakers. So if anybody here wants to give a talk at one of these or wants to volunteer somebody else, feel free to send us an email. We will contact them, potentially invite them for this seminar. So we have a lot of empty spots this year. So always looking for suggestions. So for that, you could just email any of us. We'll be happy to try to organize that. So for today, I'll tell you guys about recent methods in protein structure generation and design, but more focusing on specifically some of the methods that we actually implemented into Colab design framework. The reason I'm calling this part 2 is because I think about a year ago, I gave a different talk that also introduce ColabDesign. For this, we don't assume that you've watched that, so don't worry. So this is going to be technically part 2, but it's actually not related to that completely. So for today, the things that I'll cover is some interesting additions that we've added to ColabDesign, such as the idea that be able to predict structures, such models like ProteinMPNN, and how we could actually generate sequences, and then also some integration with some really exciting methods like RFdiffusion. So these are some things that I'll talk about throughout the day, but the roadmap that I'll follow today is sort of describe to you guys what is protein design. So I assume most of you guys know, but I think it's also good to always sort of step back and think about like the big picture. What do we mean when we do protein design? And then do some comparisons. There's been some really cool new methods like Chroma from Generate that came out, and we're like, hey, how is this different from RFdiffusion? And then more importantly, how is it different from hallucination? So these are some of the methods that I talked about last time, and something the community used to use, but I think there's sort of maybe a comeback to hallucination that's maybe coming soon. And I'll touch upon that towards the end. So this is the roadmap that I'll follow. And actually, let me see if I can hide this little guy. Yep. And as I said, feel free to interrupt me with questions. You could raise your hand or you could start yelling. It's totally fine. And if you want to grab a drink while I'm talking, that's totally fine as well. I won't be offended. All right, so what is protein folding? And I guess most of you guys know, so protein folding is this idea of trying to find a structure that falls within the global free energy. So like we're trying to find this minimum. So the idea is like, as you're searching through the space, you're trying to find this conformation. And so this is what we typically think about what is protein folding. This is how we think proteins fold inside the cell, but also when we develop algorithms inside the computer, we also try to mimic this process, like somehow define this energy function and some ability to sort of search this space. And so typically here we go from sequence to structure. Then once we have a structure, maybe we can define some function afterwards. But today we won't talk about function because we're just going to stop at structure for now. Now, protein design, which also is sometimes referred to as like the inverse protein folding problem, is going the opposite direction. Like now what we're doing is this is the target that we want. Like this is the structure that we want. And what we're doing is we're changing the sequence, like we're changing the actual sequence of the protein. And what we're hoping is to find a sequence that will fold into that structure. And I think this is a pretty critical point to think about because I think often people think like, oh, we're designing sequences or we're designing protein structures. But in reality, what we're really doing is we're actually designing conformational landscapes or maybe protein folding landscapes. Like we want to essentially find a sequence where that is just one solution. Like you could also imagine a scenario where there's like multiple peaks, maybe if you're designing a metamorphic protein. But let's say in this case, we want to design a protein that folds into this conformation. So this is traditionally what we would say is the protein design problem, is trying to find this. And the reason why this is related to protein folding is because when we come up with a sequence, then we essentially traditionally would have to do some kind of a conformational sample search to see, hey, does this sequence actually fold in that structure and no other structure? So it's actually quite an expensive computation to do. But this traditionally, Ken Dill, I think since the 90s, has been referring to this also as the inverse protein folding problem. Now, the reason I'm sort of highlighting this or emphasizing this is because there's been quite a lot of papers out there that all claim they have an algorithm for inverse design or inverse protein folding. And you may have seen all these different papers. And I think every time I see one of these, I always email the authors or tweet, reply to them and say, hey, you're incorrectly using this term because none of these models actually consider the actual protein folding problem, nor trying to think of a sequence that will actually fold into that conformation and no other conformation. And what I mean by that is this idea here is like, imagine you have a model that models the probability of sequence given structure. And let's say this is your little eyeball here and you're looking at that target and you're thinking of some sequence, like you're changing the sequence, you're hoping for the best. But in reality, what may happen is that you come up with some kind of sequence that folds into this target, but also now has the ability to fold into a completely different structure. And so if you have a method that's only modeling the probability of sequence given structure, you actually are not doing, like I would say, is a real inverse folding because you need to consider the entire conformation landscape and you need to maximize the probability of what you want and minimize the probability of everything else. So if you have a method that actually does that, then I say, okay, that's okay to call it inverse folding. But otherwise, all these methods here I think at some point I call this like the shame list, but it is a list of all these methods. And I think partially has to do with like, I think there's like one paper that called it inverse folding and then everybody else called it inverse folding after. And so to put blame on one paper, okay, maybe I shouldn't. But anyways, you could probably look up the terminology. But this is a really, really important point, and I think this is the point that I'll be kind of emphasizing throughout the talk, is that in reality, what we really care about is we want to make sure it folds into that structure, no other structure. And this is why we were really quite excited in the field when AlphaFold came out, for example, where AlphaFold can predict a structure based on sequence. And so we could quickly sort of check to say, hey, does it fold into that structure or no other structure? And so one sort of, I guess you could say, one for real's way of solving the inverse folding problem is to potentially just invert the structure prediction model itself. So what I mean by that is, like, imagine if you have a model, like a structure prediction model, that actually returns to you the structure given sequence. The exciting part here is that you're essentially looking at the whole space, right? So you're optimizing the sequence and you're automatically checking to say, hey, does this sequence fold in that target and no other target? If it's not so confident, for example, the prediction, then, hey, maybe it can go into multiple locations. But in this case, we're saying, hey, we're looking at everything and we're trying to optimize just for that target. And so that's what we say. This would be like the true inverse folding if we could ever achieve that. Yep.
I hope I'm not splitting hairs, but then we should redefine what protein structure prediction means, right? For a given sequence, you say that there's a structure, but what you're arguing is that there could be a similar conformation that might be very similar in energies, right? So would you say that we should start redefining the protein structure problem as well? Because it's all about conformation rather than a single structure.
True. So I mean, so we assume most structures fold into a single structure. Sorry, most sequences fold into a single conformation, but that's not always true, right? There's many proteins that have different conformations. There's always extreme examples where, like, metamorphic proteins that change conformations. So actually, if your goal is to design a metamorphic protein, then you do actually want like two, I guess you could say, values going down, and that's totally what you want. But also maybe you want something that it's easy to transition between two. Like if there's a huge barrier you have to go through, that's also not good. So in this case, when I say structure prediction, we kind of assume that the method, like for example, like AlphaFold, is trying to find the most likely structure, like the single most likely structure. But even that I would say is probably an approximation at best of the whole process. And so maybe in the future, we will try to actually model protein folding and maybe we'll solve the problem. But in the meantime, I would say inverting a structure prediction model is at least one step closer to that approximation of modeling the real problem. Yeah. All right. So hopefully that made sense. I'm on my second beer, so I hopefully won't screw up. I just realized these are like 10% or something. So, okay. Hopefully, okay. I'm going to try to walk through this really fast because otherwise I will start to say nonsense. Okay. All right. So let's go through an example for so here I have a protein structure, but let's say I want to design a sequence that folds into that protein structure. One way what I could do is I could take this protein structure and I could describe it as like a contact map or a distance matrix, something that says residue pairs and how they're interacting with each other. So we have this helix right here is defined as this helix here, and the interaction of these parts of the loop are interacting with the helix. You can see these contacts appearing here. There's some kind of sound in the back, but let's ignore that for now. But now let's say we have a model like AlphaFold. We can give AlphaFold some random sequence. We could also give AlphaFold some other information, such as the positional embedding. I won't go into too much detail here, but turns out what you could do is you could tell AlphaFold that the N-terminus and C-terminus are actually connected. And so then you could actually get back a structure that's actually cyclized. And it turns out it's relatively easy, like literally one line of code, and you can make AlphaFold return cyclic peptides. So in this case, I have a completely random sequence that I'm giving AlphaFold, and I'm giving it some information about how far away each of the amino acids are from each other. And I get back this distance matrix. And so this distance matrix does not look like this one. And so what I could do is now is I can compute the difference between this guy and construct some kind of loss function that just says how different they are from each other. And then I could propagate this loss all the way through the model, get some kind of gradient that tells me how to update the sequence. And so this is sort of the, I guess you say, traditional way of by traditional, I mean like 3 years ago, is the way of doing protein design is that you invert a structure prediction model, and then you try to change the sequence until it folds into the desired structure. And so here's a quick animation of showing that exact process where I'm changing the sequence, I'm updating the sequence, and then I'm updating it until it matches the desired structure. And here now I come up with a sequence. Here I'm showing as this one hot representation where this is the number of positions. These are the 20 amino acids. And you can see, for example, I don't remember which of these are which amino acid, but you could probably look it up at some point. But it turns out that that codes for some kind of sequence. But now once you have that sequence, then you can ask the question, does this sequence actually fold into the desired structure, right? And we've sort of inverted the model. And so one could argue, like, well, maybe we came up with some kind of adversarial sequence, right? Maybe we found a way to trick AlphaFold. So we need some kind of orthogonal way of testing this hypothesis that we found out a better sequence. And so one approach to do this is to take the sequence and pass it to a method called Rosetta ab initio. So this is I guess most of you guys here are probably familiar with Rosetta, but the way Rosetta works is that you could take a sequence, in this case, I just made up some sequence here. You pass it to Cypred, a secondary structure predictor. Then based on the secondary structure prediction, you could pick some fragments, like extract fragments from current structures, and then you could recombine these fragments guided by the Rosetta energy function. So the Rosetta energy function includes things like the solvation, hydrogen bonds, van der Waals, and so on. And these are methods going back to the 90s and maybe early 2000s. And the cool thing is if you do this, you get back something like this. So what I'm showing here is each dot here is a separate protein folding trajectory using a Rosetta ab initio protocol. And one thing that's really exciting here is if you use a method like this, where you inverted the structure prediction method, in this case AlphaFold, and you try to maximize the probability of structure given sequence, and then you take the sequence and you run it through Rosetta, you actually get this funneling structure. So this is essentially showing here that the structures that are closer to the solution that we want have a lower energy. But now if we use like the standard, I guess, traditional method from like 20 years ago, so that one, this one actually, you can see has potentially alternative minimas. Like we want this to be the solution, but we see that there's other proteins out there that actually may have a similar energy. So like this is something we would assume may have a trouble folding up in the cell, while this right here, we would assume like, hey, it only has one correct solution, one global optima. And so hopefully this will now fold into the structure. So this is all computational experiments. And so in collaboration with Gurov et al. And others, we've actually took this sequence and expressed it and actually got the structure with X-ray crystallography. And the structure is really, really similar. So in fact, it does appear to work really, really well. And so inverting the model, we're getting sequences that even Rosetta likes in terms of constructing this folding funnel here, I guess you could call it. And the structure itself also experimentally works, or the sequence folds into structure. Yeah, Nick?
Sorry, a couple of questions.
Yeah.
One was, could you talk just a little bit about back propagating the gradient of the loss?
Yeah.
In order to sequence is discrete, right?
Yep.
But, this back propagation, you're changing the sequence in a continuous fashion. I just want to know your thoughts about when is it appropriate to turn that not realistic sequence because it's fractions of amino acids, let's say, into a one-hot encoded sequence. That's question one. The second one is a lot of papers these days will use a model like AlphaFold and hallucination or something like that to come up with a sequence. And then you can't really use AlphaFold to predict the sequence and say it's going to predict the structure and say, oh, okay, look, it's self-consistent. So they don't use Rosetta, but they use Rosetta Fold, or they use some other kind of structure prediction. Here you use Rosetta.
Yep.
And I assume it's because it's a very small protein, and also you could probably model cyclized peptides.
Yes. Yeah.
Maybe it's difficult with other models, but I sort of want to hear your take on how valid is it to actually design up a sequence through hacking AlphaFold and predicting that structure with Rosetta Fold and saying, oh, they're self-consistent, therefore it must be good.
Yes, yeah, that's a great point. So coming back to your first point, first question about sequence. So in this animation here, I'm just showing the first steps of ColabDesign, the way we implement this is like, what we do is we initially make the sequence completely continuous, and it could be anything it wants to be. And then what we do is we switch over to what is referred to as a straight through trick, where what you could do is you could pass gradients through the softmax, but then you could force the things to be categorical and continue optimizing afterwards. So there's and if you're curious about the details, if you go to my part 1 on YouTube, I go into the details of that. But yes, so there are these, in machine learning, there are all these different tricks of forcing the sequence to be categorical. But often what people do is they don't really force it right away. They kind of decrease the temperature and try to push it towards the end to become discrete. Because otherwise, if you force it right away, it turns out like a single mutation completely changes the structure. So it's very, very unstable. And actually, towards the end of the talk, I'll tell you guys about some work that I've done with Chris Frank about trying to address that problem. And so you'll hear more about it. So as far as your second problem, the second question, so in this particular example, we wanted to ask the question, is it true that when you invert a structural prediction model that you get back a sequence that has a single global optima? And so in that case, we actually did need to get essentially a conformational landscape to some extent. And so each dot here is a separate MCMC trajectory from using Rosetta ab initio. And so we see this funneling kind of shape. So our goal was to see this funnel. But that being said, in practice, this is, as you said, is actually really, really expensive to run. For small peptides, it's actually really fast. Like I think it's like just a few minutes for each example. But for larger proteins, like it could be like 10 minutes to an hour or so, depending on the size of your protein. So in practice, it's not something we could do for every example. But also, there's been this idea that maybe instead of using this Rosetta ab initio thing, we can use an external orthogonal structure prediction methods, right? So it is like if you designed with AlphaFold, you could use like Rosetta Fold to predict, or if you design with Rosetta Fold, you could use AlphaFold to predict. Yeah, and so the assumption is that they're capturing slightly different things. I mean, they're kind of similar models, but if there's like an obvious adversarial mode that you're able to bypass that in some ways. So that's the essential idea of filtering. Yeah, back question. Yeah.
So I really like this one because I think like more papers should start showing that AlphaFold or whatever design structure they're proposing, how stable it is in the landscape. So like that, the question is that, is it the accident or is it actually a deliberate feature of the inverse AlphaFold model that we have?
That's a great question. So in this case, we say it is deliberate, like because we're assuming that the structure prediction model. So if we go back a little bit, a few steps here, we're assuming like methods like TR Rosetta, AlphaFold, Rosetta Fold are essentially considering the entire possibilities and say, okay, this is the most likely structure. Like it's looking in some way implicitly at this conformational landscape. And we do have a paper where we analyze that a little bit more carefully with TR Rosetta, saying, hey, let's generate a whole bunch of sequences with TR Rosetta, like inverting the TR Rosetta algorithm, and then doing this exact same calculation that I showed here. And we do see that models with the inverted property do have a more funnel-like shape, like when you do with Rosetta Fold. Sorry, when you run Rosetta ab initio forward folding algorithm. Yeah, there's a lot. Unfortunately, I think we call this algorithm like 10 different ways. So it's hopefully it doesn't confuse too many people. But yeah, another question back. Great question. So this is the Rosetta energy function. And I believe it's well calibrated, but who knows? Actually, if anybody else here wants to chip in, such as I know some reviewers are like, you have to call it a score. You can't call it energy. So, but anyways, it's a great question. So it is calibrated from the point of view of there's a lot of known measurements. And so you could adjust these reference energies for each amino acids to sort of match known kcal calculations. In this case, how well this is calibrated for this example, I'm not sure. But let's just assume that it's all real for now, because we do have a crystal structure and it looks fine. That being said, we have reviewers that are currently reviewing this paper, and they said crystal structure is not enough. They want us to do NMR experiments. Yeah. Now, the crystallographers, or sorry, I guess the NMR excuse is that maybe sometimes if your protein's really, really small, maybe due to crystal packing, you could maybe change the conformation. And so maybe that's the wrong structure. But we have like 10 of these, so I think we're fine. But yeah, yes. But that's a great question. So here, the point here more is to say, hey, compared to the traditional methods of Rosetta ab initio, how do these sequences actually compare? Like you actually use Rosetta itself to model, to come up with a sequence. And then if you use like AlphaFold inverted to come up with the sequence, how does it compare? And we see that there's a funneling shape, which suggests that we are indeed, by inverting the model, considering potentially the entire conformation landscape. But that being said, you guys may have heard stories that this doesn't always work. So turns out, like if you try to actually do this, the exact same procedure I just talked to you about, let's say, hey, let's try to hallucinate a larger protein, or let's try and make a protein that matches an existing structure. And if you actually take these proteins that are much, much larger, experimentally, they don't actually work. This is an example, and by experimentally doesn't work, what I mean is that it's not monomeric, and it is not either, or it's not soluble. Like the protein seems to crash and not work so well. And it becomes a kind of question, like, why is that the case? Like, why is it with small proteins, it seems to work great, but then when we start to try to do the exact same procedure for large protein, it doesn't work? Like, what's going on here? Does anybody want to guess what's happening with these large proteins, or why? Yeah.
You consider multimeric assemblies in the first building model? Because that's when we have the helical bundles, they're always monomeric or crazy structures. Multimers that maybe considered during monomer stage.
Could be, yeah, definitely. So, I mean, the thing is, the model technically may infer that this forms some kind of oligomeric assembly or part of a much larger protein or so. So that's a great point. So like AlphaFold itself is implicitly making some assumptions about the protein sequence, like maybe it's part of a larger protein because it was actually trained in crops. So there could be some of these weird adversarial modes, but definitely. I don't know if you guys have been watching YouTube shorts recently. This guy may pop up once in a while. So turns out we have a problem here. So the problem is that sort of what you alluded to, but also the fact that technically AlphaFold, RosettaFold, TR Rosetta, all these methods have actually been only trained on real data. Like what I mean by real data, like only sequences that actually do fold. So the model never really has to understand like what is a good or bad sequence, because it was always given information that essentially is always trained on good sequences, right? And so one hypothesis is that maybe what's going on here is that the model maybe doesn't even look at the full sequence. Like it looks at the whole sequence and says, hey, this kind of looks like a Tim barrel, and it predicts a Tim barrel. Or this kind of looks like a beta barrel and predicts a beta barrel. And so maybe for small proteins, when you have a really, really small protein, it's really hard to do that. Like you can't really ignore it. Like any mutation can change the structure. But when you're dealing with a large protein, maybe there are actually ways to, I say, trick AlphaFold or trick any of these methods to give you the structure that you want, but maybe that sequence is actually not that optimal, right? And so the assumption here is that maybe this is what's happening. It's like somehow we found like adversarial sequences that trick the model. So how do we get around this problem? And so one solution has been say, well, what if we somehow model the probability of sequence? Like if we somehow have a magical model that can tell you, hey, this is actually a good sequence. Yep. Nick?
Quick question.
Yes.
For all of those data points that you have, did you fold the sequence with Rosetta fold? And did it still fold?
So all of these points, I forgot to mention, but all of them, so AlphaFold loves all these sequences, which I guess, yeah. I don't think Rosetta Fold 2 was out at that point, so not at this point. But yeah, that's something we could try again. Yeah, that's a good question. No, I haven't done that. I haven't done that yet.
Does this suggest the potential of negative reinforcement learning with the millions of garbage sequences that the Rosetta community has produced over the years?
Well, if somebody makes a database of those sequences, so unfortunately, there's not too many. I mean, I guess you could always make negative sequences, right? You could put like hydrophilics in the core or something, right? But yes, definitely. I think one potential solution around this problem is maybe say, well, maybe we could reformulate this problem as some kind of contrastive learning, right? We could say, hey, we got bad sequences, good sequences. The model should not return good confidence structures for bad sequences. So that could be maybe one way to get around this problem. Or like as I'll show in the next slide, is maybe you have some model that models a probability of sequence. And so one way to get around this problem is to use ESM. So ESM is a protein language model. And so here, what I'm showing here is the same structures, the exact same structures. It might be a little hard to see here, but here it's showing the probability coming from AlphaFold. So AlphaFold really, really loves all these sequences. But one thing that I want to highlight here is notice there's a bunch of red here. This red is regions that the language model hates. So the language model actually really hates this sequence. And so one idea, this is in collaboration with Alex Rivas at the time Facebook, now Meta, but now I think Evo scaling. So there's been a few transitions here. But the idea here is that, well, what if we have this structure prediction model? In this case, this could be AlphaFold, or in this case, we could just take, for example, language model, take all the attention matrices, stack them together, and train a very, very simple model, just literally a linear layer that returns a distogram, like the distogram, the distances distribution for every pair of positions. And so we essentially have within, and I guess you could say this is an earlier version of ESM fold. Before returning to fold, you just return a distogram. So we have a way to model the probability of structure given sequence, but also just from the way language models are trained, we also have a way to actually model the probability of sequence. So what I mean by that is you can mask any position, and then you can have the model predict the probability of those masked positions, and then you can ask the question, how well does your sequence mask those predicted probabilities? So essentially, we have a way to score the sequence, and then we have the way to score the structure given the sequence. And if we maximize both of these probabilities, and so this is what was done in this paper here, language models generalized beyond natural proteins, is that in this case, we now do a joint optimization of both these objectives. So both optimizing the probability of sequence, but also optimize the probability of structure given sequence. Which I believe, if the statistics are correct here, models a joint probability distribution of those guys. And so then when you do this, suddenly all those designs start to work again. Like, so now experimentally, as far as size and solubility and so on, the structures look to be, you're getting good proteins. Now, of course, the structures could be completely wrong, but we assume that they're correct. So this is ongoing work. Unfortunately, wait, this is on YouTube, so I can't probably say this, but yeah, ongoing work. So I'll just come, we could discuss this later.
Sergey, can I jump in here? This is Guy Monteleone.
Yes,
Can
yes, of course.
can you calculate also, the likelihood of the dimer, the likelihood of the trimer, and use that as some sort of screen also on which designs are likely to form monomeric well-behaved systems?
That's a great question. Yes. So in this case, we're only optimizing for single structure, right? But we could do some kind of negative design, right? Say, hey, make sure it's not a homodimer, not a tetramer.
Because AlphaFold and RosettaFold do a great job on homodimers and homotrimers, and you may be able to filter that way. Yes,
that's a great point. I don't think it's currently done by anyone that I know, but that's a really good suggestion. Maybe we should add that in for the next round. Yeah, thank you. I don't know if you guys heard, but Guy was suggesting that maybe we should, maybe alluding to the comment made by Nick earlier, that or actually not Nick, somebody else made the comment earlier, that maybe some of these didn't work because they form homoligomers, right? So we could directly optimize against the homoligomer structure, right? So that could be an interesting loss function to add to minimize that. It's a great question. Okay, so these guys now appear to work, which is really exciting. But you might wonder, it's like, well, both of these things that I just told you about, it's like inverting AlphaFold, inverting ESM, is really, really, really expensive, right? It's like, this is, I think for this calculation, I believe they did like 50,000 runs of ESM, which I think no one can actually do, except unless you're in Facebook. But I guess, unfortunately, those resources have been diverted to all this metaverse stuff. So we can no longer do those kind of calculations anymore. So one question you might wonder, this appears to work quite well, but is there a better or faster way of doing this? And I think one of the really exciting areas of research that have been coming out recently is this idea, say, well, maybe instead of inverting a model, instead of using a model like an energy-based model in optimization, maybe we could actually have some kind of model that directly generates structure or directly generates sequence like maybe use some kind of diffusion like process, right? And so, and this sort of comes from this idea that maybe you could train a model that takes, for example, a cool picture of a dog, and then let's say add some noise to it. Like you could just add some noise to it, and then you could train a model that sort of undoes that noise, right? So you could progressively add more and more and more noise, and then train a model that sort of undoes that noise one step at a time. And so then the cool thing is now you could start with completely random noise, and then actually completely go backwards and maybe get a picture of a dog, right? So that would be kind of cool. So this is research. It's been going on for quite some time in the image world, and this is how things like DALL-E, Sora, I think, also does something like this with the videos. But the cool thing is that you could actually do the exact same thing with proteins, right? So you could start with a structure, add noise to it, and then unnoise it, add more noise to it, and unnoise it. And then later on, you can start with completely random noise and take iterations and then essentially denoise until you finally got full structure. And so this is maybe an alternative way to do this things much faster. Like we quickly get a structure. Maybe we could add some conditions to this to get it to satisfy some kind of conditions. But once you have this, the question is, what do you do next? Because unfortunately, what this does is it just gives you the structure. That's all it does. And so in this particular example, it's taking the Rosetta fold model and fine-tuning it to remove the condition of sequence. And so literally all you're doing is you're just feeding the coordinates back over and over and over through the model. And you're starting from random noise and you're denoising to get a structure. But this only gives you the structure, nothing else, right? So then how do you actually put a sequence back onto this model? How do you get a sequence, right? So that's the question. So it turns out one way to do this is to use a method that is actually trained to do the opposite, right? Given a structure, will return a sequence. One example from really, really early work from 2019, I guess it's not too early, but it's relatively early. So this method essentially takes a structure, breaks it down to the edges and nodes, and then goes through a layer of encoding the structure, and then goes through a layer of decoding the structure to return the sequence. For this purpose of the talk, don't worry about understanding these blocks. The point is you get a structure and you get a sequence out. So sequence, structure, and sequence out. But what are exactly the features that it uses? So in this case, the ProteinMPNN method, which was developed by John Ingraham, is it takes a structure and decomposes it into essentially a series of distances, but also the orientation between each of the residue pairs. So that's the features that go to the edges and nodes. If you sort of stare at this, you might start to recognize there's some features related to Rosetta, because Rosetta itself is also composed of pairwise terms and also considering like the orientation, the distances between each residue pairs. But one thing that's really, really exciting here, and I think this is one thing that I believe makes it better than methods like Rosetta, is that it has the ability to integrate information not by just the direct neighbors, but maybe multiple shells of information. So what I mean by that is like you're looking at this position here, and you're encoding the information at this position, giving all its neighbors, but then you do this multiple times iteratively. So you get the neighbors of the neighbors of the neighbors kind of passing all the signal all the way to until you get to this point here. So the idea is like you could potentially learn some more complex energy functions besides just maybe some linear pairwise decomposition that we use in like traditional energy functions. And so this model gets integrating a bunch of information at every single node, but then afterwards you can then run this model as a decoder and say, okay, now given all the information that I've placed at every single node, let me decode and generate some kind of amino acid at that position. So now you can model the probability of this amino acid given all the amino acids you sampled up to that point, but also the rest of the structure as well. So this is the idea of ProteinMPNN. Any questions? Yeah.
What does this do to the concept of explainability? But you just say energy is my ground truth and that explains everything.
You're wondering like what energy you actually learned, or maybe try to figure out a way to extract this energy?
No, I mean, so you're doing all of this and this informs, say, that and that amino acid should be in that position. How do you explain that that is the right answer? Do you use energy as the proxy for the right answer?
So in this case, we're not explicitly modeling any kind of energy. We're just modeling the probability of that amino acid given all this information around it. Now it's possible the model learns some version of energy when it's, so the idea is like you could think of a probability distribution of some kind of like a Boltzmann distribution, where you're considering all possibilities of that state or that site. But the model itself doesn't explicitly do any kind of energy calculations. Maybe implicitly learn some kind of energies. But the key part that I'm trying to get at here is that traditional energy functions are essentially say, well, for a given atom, look at all its neighbors. And then based on that, you do some kind of pairwise decomposable energy function that just calculates maybe some distances, maybe calculates some intervals. But one really exciting thing about these message passing neural network, and sorry, I should have brought this up, message passing specifically, is that you're instead of just looking at the neighbors, you could pass the neighbors of the neighbors of the neighbors of neighbors and integrate information about the entire environment and the whole protein. So that position now has information about the whole protein, not just the immediate neighbors. And so one possibility would be like, maybe something like this might be a better job at capturing like hydrogen bonds that may be something that's hard to decompose into a pairwise parameter. Like for example, like you can't have like 1 acceptor and 10 donors, to give you an example. And so maybe this would be say, hey, actually, given the whole context, there's only one possibility here. But whether or not it actually does that, it's a great question. I wasn't planning to show this, but since you brought it up, so we have a notebook. So if you guys are curious, we have a special notebook that takes a protein language model and can actually extract the conservation and the coevolution directly from the model, like any kind of pairwise potential, site-wise potential. And so if you have a favorite protein, in this case, let me just run this. Maybe I'll show you guys in a few slides while this is running. But you could take a ProteinMPNN model, give it a structure, and say, ask the question, hey, what amino acid probabilities are you getting at every position? And then for every pair of positions, you could ask the question, what does that 20 by 20 matrix look like? Like if you put one amino acid here, what should be the other amino acid here? And that might be some version of an energy based on, if you believe Potts models are real, and then stuff like that. Okay, but we'll come back to that in a second while it's running in the background. Okay, yes.
So at the start, you gave us this really nice principled argument for inverting protein structure prediction models, because that it's viewing the entire landscape or what the model knows about the landscape. But I don't think that same argument follows, right? Because even using MPNN on top of a given structure, it's still just conditioned on that structure. So
definitely. It's I'll show a few slides to show you guys how that's corrected for. Yeah. Yes. That's a great point. So so far I've been telling you guys how it's bad to use the probability of sequence given structure, or we're using the probability of sequence given structure, or vice versa. But what I'll show you guys in practice, we still use both approaches and combine them together to try to actually model, I guess you could say, the real inverse folding problem. Yeah, but oh shoot, there's like people talking here. All right, I think I'll come back to that later. Okay, so it's too much to absorb. Okay, so so this is the ProteinMPNN that was developed by John Ingraham, but unfortunately, when the code got released, there was no model weights that were available. Like the code just got dumped online, but the code was dumped online, so that was great. So that's a first step to recovery. But now the question is, how do you go beyond that, right? How do you actually be able to use these models? And so Hustis, at the time was at David Baker's group, took the ProteinMPNN model and said, okay, let's retrain that model so we actually have the weights. But on top of that, said, you know what, actually, I could add more information to this model. Instead of just thinking about like pairwise distances, maybe I can compute the distances between all possible atoms in the backbone. And it turned out that actually helped quite a lot. But on top of that, you say, well, maybe you have 2 proteins. So instead of just training the model on a single protein, you can train it on multiple proteins. And this becomes quite important for the question that I think Guy brought up earlier, that, well, if you want to optimize the protein to not, let's say, have hydrophobics on the surface, unless you explicitly add all those other units, there wouldn't be an incentive to put hydrophobics on the surface. So essentially now we have a way to explicitly tell the model, here are 2 proteins, come up with a sequence. But then if you just give it a single protein, then the assumption is that it's like, hey, it's a single protein, I'm not going to put hydrophobics. on the surface, it's a self-contained protein. So now we have an explicit way to define this information. And I think this is a really important point, because the original model was essentially just training on SCOP domains. But these domains are part of a much larger protein. And the problem there is that based on those statistics, you're like, hey, yeah, it's totally fine to put hydrophobics on the surface, right? Because maybe there's another protein out there somewhere or another domain out there. But now by explicitly training on fold complexes, you're able to capture potentially the fact that you're explicitly adding that information to the model. So that's the motivation of why to do even if you only care about monomers, it's really important to train on multimers, because you tell the model about the whole environmental context. Okay, so to get back to Hannah's question and say, okay, well, cool. So you have a way to go from structure to sequence. Are we done? I would say that is not the end picture here. Because then what we could do is you could take the sequence now and then pass it to AlphaFold and say, does it actually predict back the structure? And in practice, no one runs this method alone and no one runs this method alone. These methods are run together simultaneously. So the idea here is that, yes, we want to come up with a sequence, but then we want to also make sure that the sequence folds into that structure and no other structure. And so you could jointly optimize these objectives together, or you could essentially just generate like, I don't know, millions of sequences and then run them all through AlphaFold and then test for that ability. So it's like you could, and essentially what I guess get the claim here is that you're essentially doing the same thing, right? You're either generating a bunch of sequences and you're scoring them with AlphaFold, or you can include AlphaFold as part of the objective. Which way is better? I don't think there's any reason to think one or the other is better. It's just maybe it's faster to generate sequences first and then maybe screen them with the other ways, so on. Right. So at the end of the day, we're still doing this, I guess you could say, inverse folding by combining these two approaches. Like we want to make sure we find a sequence and we want to make sure the sequence folds back into the same structure. Does that make sense, Hannah? Did I answer your question? Yes. I don't know if that. No comment. All right, maybe I'll try that. Okay. No. All right. So let's.
Sergey, I have a question.
yes,
So
yes.
about diffusion, which is that if I know the structure I want to make, let's say I have a peptide and I want to design a protein, I want to design a helix that binds into a groove on the peptide. Well, then I wouldn't use the diffusion in that case, right? I just want to make the sequence that makes that peptide. I would use the diffusion approach if I needed to find a structure that binds in that cleft. Or is that naive?
Oh no, that's totally right. If you already have the structure that you want, you don't need to do any kind of diffusion. You can just go directly from structure to ProteinMPNN to design the sequence, but then you can afterwards check to see, does the sequence fold back into that structure?
Yeah, absolutely, that part. But the diffusion is to create a scaffold essentially that's going to have a feature that you're looking for.
Exactly.
Right?
Yeah. So for those who didn't hear, Guy was asking, let's say if you actually already have a structure, do you need to do diffusion or anything? So the answer is that no, you don't have to do any kind of diffusion if you already have the structure. That being said, there has been some work by Susanna and David Baker's group that showed that you can actually do something called partial diffusion. Like you could take an existing protein interface and just add like a little bit of noise to it. And often that little noise could make the structure just a little bit better. And so maybe you found one solution. So the problem is with ProteinMPNN is that if you give it a structure, it's often will give you very, very similar solutions every single time. But if you actually run diffusion on existing structures, but just do a partial, what I mean by partial is instead of fully, let's go back a little bit, like instead of fully denoising or noising the structure, you could just take maybe a few steps, add a little bit of noise and then denoise it. And Susanna, I believe, found that when you do that, you can actually get much better sequences. There's a preprint on this, I believe.
So it can't be
Yeah.
just add the structure or possible can be different sequences.
Well, if you add noise to it, it's not, I don't think that's a great idea, but what you could do is you could add noise to it and then denoise it. And so when you denoise it, it may actually improve the structure a little bit. Later on, I'll show you guys some slides. Let's see how much time. Oh, shoot. Okay, I think we're running out of time. But I'll show you a few slides later that it definitely helps to take a structure and add a nil noise and then denoise it. because you might be able to fix little details, like maybe the strands are not paired properly, maybe the peptide is not perfect. I mean, if you're dealing with crystal structures, maybe you're fine. But if you want to sort of create like an ensemble of sequences, you could definitely use that kind of approach. And I'll show you guys how to do that later. I think we're running a little bit out of time. So if anybody needs to escape because they have like more important things to do, don't feel free. But I guess I'll just keep going at this pace. Is that okay, people? Okay, yeah. But if anybody needs to escape, don't feel I won't be offended at all. So just yes. I'm still half this. I have to take it slow, otherwise I'll start making up things or hallucinating things. All right, so so the big picture here is exactly what Hannah sort of alluded to earlier in the question, if I understood the question correctly, is that and sort of the point that I was trying to make throughout the talk is that at the end of the day, we're still trying to do the full objective, right? We want to come up with a sequence. We want to make sure that sequence only folds in that structure and no other structure. And all the designs that have been successful up to this point have essentially passed this filter. Now, you might get lucky and then maybe the sequence will fold into that structure, but in practice, what has been found is that sequences that pass both of these filters, essentially this filter by saying, hey, take the sequence, predict the structure, does the structure match my original goal? Those sequences often work in lab. And I think depending on the task, I think people are reporting anywhere from like 40% to like 10% to 100%. But yeah, it depends on your task. So it could be anywhere from 0 to 100. But nice. Okay, but let's just go through that process one more time just to see what I mean by that. So the idea is like, let's say you have the structure now with coming from RFdiffusion. You pass it to ProteinMPNN to get the sequence. Once you have the sequence, you pass it to AlphaFold. And AlphaFold returns a structure. Now you can stare at the structure and say, hey, does this look like my structure? And if it does, then you're good to go. You've sort of satisfied all the conditions that I've laid out for you guys. All right, so for the next couple of minutes, I just wanted to quickly show you guys that everything I just talked to you about is available in Google Colab. And it's supposed to be relatively easy to run. So I'll just quickly show you guys what I mean by that. So this is a RFdiffusion notebook. And we have implemented inside ColabDesign. Let me just zoom in a little bit so you guys can see details here. But you could start staring at this and maybe it doesn't make sense. So you could scroll all the way to the bottom. These instructions are actually at the bottom instead of the top. I initially had the instructions at the top and then it kind of looked busy. So, but you could scroll to the bottom, look at the instructions, and scroll back up to the top, and then you'll understand what's going on here. But in short, what's happening here is that you can say, hey, diffuse a protein of length 100. That's literally all you have to do is type the letter 100 and it will give you a monomer. You can say, hey, diffuse a heteroligomer of length 50 and 100. So 50 colon 100. Or you can say, hey, I want to make a homodimer. So you could say diffuse 50 and then cyclic symmetry order 2. And so this is like completely creating new proteins from scratch. If you scroll up to the top here, you'll see those options. Like this is where you define this context here. So this is for completely unconditional diffusion generation of new structures that maybe don't exist in nature. But let's say if you have a more useful application, besides just making cool looking proteins, unless you're an artist, is you say, well, actually, maybe what I could do is I could start with some known protein, in this case, some random protein 4N5T. And you can say, hey, I'm going to grab chain A from this protein, and I'm going to diffuse a new binder to it of length 50. So you could specify a PDB code. So essentially what will happen inside the code is it'll take that protein, and then it will initialize some random noise of length 50, and then that noise gets denoised until it becomes a binder. And so I won't go through all these options, but I just wanted to show you that this context is quite extensive and that you can say, hey, grab a chunk of one protein and diffuse a length of 70 to 100. Or maybe instead of binder, maybe you want to fix a motif and diffuse the rest of it. And so all these options are available here. So one suggestion, if you guys want to come back and try this out at home, is to go through these examples. and then try to put your own example in and try to see what happens. And so all these options are, I think, relatively well documented here, hopefully. But just to go through, you guys, one example is, in this case, I said contegs 100, so it's going to diffuse a protein of length of 100, and I'm going to take about 50 steps. In 50, it turns out about 50 is enough for most objectives. Sometimes it helps to improve to increase this. You can specify your symmetry, and so then in this case, if this was live, this would be like a video showing the diffusion process you're running through it. And once you're done, you'll get back a structure. So in this case, here's your structure. But just what I just said to you guys, that is not enough, right? So all the diffusion does is just gives you the structure. So now we need to put a sequence on that structure, right? So what you could do now is run the next cell, and the next cell will actually run both ProteinMPNN and AlphaFold, right? So the key parts here is that it generates like 8 sequences and then passes each of those 8 sequences back to AlphaFold and asks the question, does the structure predicted by AlphaFold match my structure? And so here, these guys are in this case, reported by RMSD. And so you can see it's definitely like this sequence did not fold back into the structure that we wanted, but there are other structures that did fold, other sequences that fold into that structure. So here you can see here's an overlay of the design protein and then the protein that was predicted by AlphaFold with that sequence. So now you can go at this point, if we had any endorsements, we could have said which company to send the sequence to, but I guess we don't know that yet. So supposedly now you have the sequence, you could express it, and in this case, we sort of passed through this whole, supposedly this entire inverse folding protocol, right? We came up with a sequence and we made sure it actually folded in that structure. And as you can see here, not all the sequences actually folded into that structure. So like this guy may have folded into a completely different conformation. And so this is the guy I would say this is probably a landscape, doesn't look like a valley. It probably has like multiple valleys or something, right? But unfortunately, we can't see those values with AlphaFold at the moment. But we could sort of just assume that because it didn't predict the right structure, there's probably multiple alternative minimas, and so it probably got stuck somewhere. And so that's the best way I guess we can make at this time. Yep. Yes. Yes. So if we do binder design protocol, what will happen in that case is that AlphaFold will be given the structure of the target protein, and then the binder itself would be told to be predicted completely from scratch, just from that sequence alone. And then you can check to see, does that binder, first off, does it fold into that structure, and then does it bind to where you hope that it will bind? So the test will happen also for the binder protocol. The one thing that I alluded to earlier about from Susanna's work is that you could also do this partial diffusion protocol. So let's say I have some structure and I just want to refine it, maybe get alternative sequences for that same structure. You can run this protocol here. So in this case, if you don't define a context but just give it a PDB, it will add noise everywhere. Or if you just say, hey, keep these guys fixed and remove everything else. Or let's say, for example, let's say if you want to optimize an interface, you could say, keep the chain A fixed and then denoise everything else in that complex. And so and one thing I should emphasize is this notebook was intended as a way to sort of troubleshoot all the different parameters. But once you sort of figure out which parameters work, you can then, of course, install this locally on your own computer or your own cluster and then run it for like thousands and thousands of times. But this is meant more like just to troubleshoot. You could also give Google money and then you can run it for days on end. So that's, you should mention me in when you register. I don't get any commissions, but if I did, that would be a good way to get rich. Okay.
Sergey, can I ask a quick question here
course.
about the protocol? So it seems something that's missing is the reliability scores, and the PLDDT scores also relate to flexibility, and there's more and more coming out like that. And so what do you typically pay attention to that? Is it just above a threshold, the PLDDT is enough, or is it used in a more nuanced way, or shouldn't one use the PLDDT in a more nuanced way to evaluate the different designs?
That's a great question. So for those who didn't hear, guy was asking, should RMSD be the only metric that we should be selecting for, or should we also consider these other metrics that come from AlphaFold? And I would say, yes, definitely should consider the other metrics, because you could have a scenario where the RMSD is very, very low, but maybe alpha was not too confident about the interaction. And so traditionally for protein-protein interactions, I believe the cutoff is if anybody knows, I think it's a 15 or something like that. But yes, so in this case, only the PAE is reported. But if you actually do the same exact task for a multimer, or let's say 2 proteins, you'll also get back an IPAE value. And I believe people in the Baker lab usually use a PAE value of about, I believe, either 12 or 15. I don't remember now. Does anybody remember? 10? Okay, yes, 10. Well, I mean, the lower the better. So I guess if you have to, you can use 10. But yes, Guy, that's definitely a good point. Yes, so RMSD is like the easiest one to filter by, saying, hey, does it recover the same structure? But if you have multiple things with similar RMSD, you might as well pick the one that is the most confident prediction. And so here we report multiple confidence. Like we report the confidence from MPNN, the lower the better. We also report the PLDT, the PTM, the PAE values. And so they're all here.
Yeah, I think there's something deeper. I think, and it's still, I think the jury's not out yet, but the PLDT measures flexibility. And that might be something that, of course, it's reliability somehow, or confidence. But also, I think we'll see more of that. And anyway, I just bring that up as something that really should be considered in assessing different designs.
Sounds great.
Yeah.
Thank you. Yeah.
You don't put or define the hotspots. Does it come with like a default hotspot condition?
So if you don't define the binus, the hotspot, the noise is completely initialized just at the center. And so if I remember correctly, I believe the hotspot tells you roughly where to initialize the noise. And so then but then I believe there's also some added conditional information there as well. So then it tries to actually place it in that location. But that detail, I don't remember exactly. But yes, so in this case, yes, if you don't define the hotspot, the noise is just initialized at coordinate 0, 0, 0. But the structure itself is already centered. So it's roughly centered there. It might be worth initializing the hotspot if you have a really, really large protein, right? Because maybe no matter amount of steps that you take, you'll never sort of move in that direction, right? So that might help to place in that location. Good question. Yeah.
For smaller protein, you can actually use this kind of binder to find that topic hotspot.
Potentially, yeah. I don't know if anyone's benchmarked that or not, but that could be the next presentation. Maybe you can give that next presentation next time. Okay, so I think I won't go to too much more details here, but essentially, all those steps that I described to you guys are all implemented here. And the main takeaway I want you guys to get from all of this is that it's very, very important to consider this whole cycle here. And so there's been actually a lot of methods, like all those inverse folding methods that I talked about, that do this first step. And then there's been a lot of structure prediction methods as well. So both of these methods will change a lot, but I think this whole idea of testing for this, essentially this inverse folding problem is probably going to stay the same. And it's been around since Rosetta Fold or Rosetta days. Like you could think of this as like Rosetta design and this as sort of the ab initio structure prediction. And now we've been changing these methods with faster and faster methods, but the same steps are still the same. Like we still want to make sure that that's the structure that sequence codes for. And that's what we hope these models are capturing. Okay, so I think, so some of these slides, I'll just kind of skim through really quickly, but the idea here, and I've already kind of described this, is that you could start with symmetric noise. You could start with noise near the binding, the hotspots, and then you could diffuse a binder essentially. You could also fix a motif and then start with some random noise and then essentially diffuse the rest of the protein. The motif could be more complicated where it could be symmetric, and so you could diffuse the rest of this. And all of this stuff is implemented in the notebook. So if you go with all the way to the bottom, you guys could try these out. This is just a really cool animation. I don't remember what I was going to say about this one, but it's but yes. Great. Okay, cool. So turns out that turns out that you can indeed, when you pass through all these protocols, like you design the sequences, they're predicted by AlphaFold. You can then take these proteins and experimentally characterize them with cryo-EM in this case, and they do appear to form the correct structure. So looks like everything's working. But you may say, hey, you know what? This seems kind of crazy. I want more control, right? Like I want to tell the protocol like what the secondary structure I want, what topology I want. And so sort of inspired by Martha's work, this idea of sort of, hey, we can run diffusion, but we can maybe tell something to the method what we actually want. Like maybe we could define like an adjacency matrix that describes the topology. And unfortunately, this protocol is a blog post and the code was never released. And so researchers have since then tried to implement this inside RFdiffusion. So now in RFdiffusion, you could do this exact same thing. You could define an adjacency matrix describing these blocks and then diffuse a protein that matches that. And we also have a Colab notebook that implements this exact same procedure. And so if you click on here, this is the C diffusion fold condition. And here you have the ability to specify the number of secondary structures. You could extract a blueprint directly from a PDB structure and then condition on that diffuse a structure. You could even take this step further and diffuse something based on the condition that binds to an existing protein. And so unfortunately, since we don't have too much time, I won't go into the details, but I just wanted to show that we do have notebooks that can do these things, and you can go all crazy and design something like this, or a tin barrel, or in this case. Okay. Okay, so, but now you might wonder, okay, so cool. So now there's RFdiffusion. There's something called chroma out there. There's hallucination that I just told you guys about. How are all these methods different from each other? Are they actually doing something different? Is there actually an advantage of one method versus another method? So I'll spend the next maybe 10 minutes just describing those. And once again, if you need to run away, don't worry. You won't offend anybody here. So chroma is one method that came out, which I thought was really exciting from the Generate group, is instead of starting with completely random Gaussian noise, what you could do is you could essentially take a protein and essentially corrupt it by preserving the polymer, essentially. So you can essentially just move the dihedrals, if I understand correctly, and then diffuse in that space. This creates an interesting way of sort of denoising and unnoising, because I think what this allows you to do is maybe make more complex motions, more coordinated motions. So instead of just moving atoms around independently, maybe you could like move an entire beta strand by just simply changing one dihedral somewhere. And so it's super fast, doesn't require Rosetta fold. There's been other methods related to this, like diffdoc and so on. But this is the method that was experimentally validated, so I'm going to talk about this. And so I have a friend, collaborator, Christopher Frank. We've met on Discord. And so Chris was like, is chroma better than RFdiffusion? And so I was like, well, let's figure out, right? So how do we test these methods? Like all these there's like essentially a new diffusion method coming out almost every month these days. How do you actually evaluate which method is better? Turns out there's a relatively simple way to do this. And this is referred to often as the backbone designability test. And so the idea here is that you could have some method that generates the backbone. So this could be a hallucination method. This could be any of your favorite diffusion method. You construct a structure. Then you could take the structure, get a sequence. In this case, you could use like ProteinMPNN to get the sequence. And then you say, does this sequence then return a structure that looks like my original structure? Now, the reason why this is a designability test is because you can imagine, let's say if you have a really, really crappy structure generation method that essentially puts all the helices really, really close to each other. What you'll do is you'll put like glycines everywhere, and the glycines will never fold back into that structure. So essentially you could say, how good is this backbone by doing this test? So like if your backbone is really, really good, you'll come up with a really good sequence, and then that sequence will fold back into that structure. Now, if your backbone is really, really bad, you're going to put glycines everywhere. If let's say your helices are far apart, you're going to put, I don't know, arginines everywhere, and then that probably also won't fold into the right structure, right? So this turns out to be one potential way, and this is referred to often as the designability test now in the literature. And so now we could do this test. And so here now we could do a competition between the two methods. So what I'm showing here on the left side is this is purely diffusion, like free, no conditions, just trying to create proteins at different lengths. So then you can use like RF diffusion, get a sequence of protein and PNN. And in this case, we could pass that sequence to ESM fold. We purposely use ESM fold and not AlphaFold because later slides I'll show you guys, we're actually going to also use AlphaFold in this test. And so we didn't want to create that extra, how do you say, bias. Yep, Nick.
Sergey, when you generated those structures with the two methods,
Yep.
did you cluster them by structural similarity? Because I could imagine one way to cheat on this is for RF diffusion to always give you a 4-helix bundle and the same one over and over again. And of course, you're going to get good sequence for that, and then maybe Chroma's giving you way more diverse structure.
That's a great question. So unfortunately, I didn't do that analysis yet, but visually they look different. They're not all helical bundles. But yeah, but that's definitely something we should definitely do in the future. But in this case, we're like, we're going to trust that they're diverse because both papers claim that they have diverse backbones. So we're going to assume that they didn't lie. And so in this case, so the RF diffusion protocol is, as I said before, you run RF diffusion, pass it to protein and PNN. In this case, instead of using AlphaFold, we're just going to use ESM fold to predict them. And so what I'm showing here is that as the protein increases in length and size, the model has a harder and harder time to reconstruct the protein. And this could be ESM fold's fault, or this could be somebody else's fault, so not quite clear. But then if you compare this to Chroma, the interesting thing you see here is that it does seem to maybe do a little bit better at length of 1,000, but these are like distribution of dots, and so it's not maybe statistically significant there. But overall, it seems that RF diffusion seems to be doing better for this task. But the interesting thing is if you say, you know what, so Chroma has its own sequence design method, essentially takes the structure and returns a POTS model internally, and from the POTS model, essentially generates a sequence. And so one question was like, well, what if we just take Chroma structure, just the backbone, and just give it to protein and PNN, just to make it more a fair comparison? And one thing that was kind of surprising was that if you take the Chroma backbones from this Chroma generation method and pass in protein and PNN, it seems to do really, really bad. It seems like protein and PNN really, really hates these Chroma backbones. And it's like not clear why. I suspect maybe, so one little detail that I didn't get into is that Chroma design, the protocol to generate sequences, were actually was trained with the information, was actually trained with noise backbones. So essentially, even if you have really, really bad backbones, it might still be able to give you really good sequences. While protein and PNN was also trained with corrupted backbones, but those backbones were only on the Gaussian level, like 0.3 Gaussian noise. And so the reason why I thought I'd bring this up to you guys is because there's been a lot of these new diffusion methods out there. And if you were to just run the standard protocol, putting a sequence on there and using ESM fold, you would say these backbones are really, really bad. But it turns out that how you actually put the sequence on a given backbone does also matter and changes the results. But one interesting thing was if you take the Chroma backbones and you do the partial diffusion thing, like you literally take the backbones and you add a little bit of noise, and then you denoise it with RF diffusion. This is just 10 steps of noise. It actually gets much better. So you can see, sorry. So this is protein and PNN, adding noise, not adding noise, adding noise, not adding noise. So it turns out that you can take a Chroma backbone and fix it, like repair it a little bit, and now protein and PNN loves that backbone. In fact, you can even add a little bit more noise, and it loves it even more, and it seems to even love it more than the RF diffusion backbones, which is kind of cool. So I suspect maybe some future protocol of all this might be to run Chroma to do some kind of global sampling, and then maybe using RF diffusion to fix all the little details. So then now the backbone looks great, and so then you can use things like protein and PNN to come up with a really good sequence. You might wonder what exactly is RF diffusion do in this case. So this is a quick example. So this is the backbone generated by Chroma from Generate Group, and you could see that it's actually not pairing the sheets quite nicely. You can see this sheet kind of has a really shitty interaction here. So then the idea is like if you add a bunch of noise to it, then you could probably fix that interaction. And so this is the kind of motions that happen when you add noise and denoise it. And I think it totally makes sense. Like there's probably, when you come up with a sequence, it's really, really probably not very specific to that interaction. Maybe that whole data sheet can move around a lot. And now by extending these sheets and essentially just switching them around, you get a much better interface between the secondary structures. And so this is an overall summary slide that overall it seems that RF diffusion is doing better. But if you actually combine all the methods like chroma plus RF diffusion, partial, and then add NPNN, you could actually do even better for these much larger lengths. So I think there's, even though I'm saying chroma doesn't seem that great, I think there is probably a future protocol that sort of combines sampling in different spaces and sort of creating structure. Shoot, okay, I forgot I have a whole nother section here. Okay, so Chris Frank, he's like, but is it better than hallucination? And so I'm just going to show you guys a few slides. Given we don't have too much time, I won't describe exactly how hallucination works. If you guys want to learn about that, you can go to my part 1 lecture. But so I'm going to skip how hallucination actually works. But one thing that's really, really exciting here, and this is something that Frank actually figured out himself, is that when you do this hallucination, and coming back to Nick's question, is like, how do you actually optimize the sequence, right? So you have to figure out a way, how do you add this gradient back into this sequence? So essentially, let's say you want to make this contact map or this histogram as confident as possible, right? And so you're changing the sequence, trying to make it as confident as possible, and you're just iterating this process. But you could do MCMC, like just randomly change the sequence and hope for the best. But what if, how do you actually add this gradient in? And so traditionally, the way we did this was to say, well, what if we do some kind of thing where we get the gradient, we take the argmax of that, and we keep adding that sequence. But the problem is, if you're moving discrete space, it's actually really, really hard to optimize. Like a single amino acid change can completely disrupt the whole structure. And it turns out it takes really, really long time to converge. But one thing that Chris Frank noticed was that actually, if you say, you know what, I actually don't care what the sequence is. Like, it doesn't have to be discrete. Like, completely don't care what the sequence anymore is. We'll just let it be negative, let it be positive. Turns out it can quickly converge onto a structure that actually looks really reasonable. And so like on the left side, you could see it's not very confident. I'm coloring here by confidence, but on the right side, it's like very, very, very confident, right? And so, and the reason why Chris was suggesting this is because Chris is like, well, we have ProteinMPNN now, like, right? We really care, all we care about is just generating a structure. We can just take this structure now, just give it to ProteinMPNN and have ProteinMPNN come up with this discrete sequence. Maybe we can use this profile to bias MPNN somehow, but that was the idea. So the idea is like, we're going to move in this continuous space to generate a structure, pass the structure to ProteinMPNN, and then give this ProteinMPNN sequences to ESM fold, and then check to see how good these structures are. It turns out if you do this, here's where this curve falls into place. So it turns out that hallucination with ProteinMPNN is actually able to come up with much better backbones at larger lengths. If you're dealing with small proteins of length 400, I guess 400 is still kind of large. Maybe it doesn't matter too much which methods you use, but it turns out the hallucination approach is actually still doing better for much larger lengths. And so this is but that being said, it looks like this line is catching up. So maybe a few more iterations on this, maybe this line will catch up, and so then we could use diffusion. So this right here is very, very slow to run because you're essentially inverting AlphaFold and you're running it iteratively, while these methods are really, really fast, but they're slowly catching up to hallucination. So Frank, Chris Frank, I mean, and his group have actually went back and actually tried to experimentally characterize some of these designs. So some of these are just characterized by SEC, other ones by CD. And in this case, they have a crystal structure of the protein. And most recently, this is something he actually just sent me like 4 hours ago, is they got a TEM image of the structure. So this is a protein of length 1000, and so it kind of looks right. So we'll have to see. So the claim is that maybe by the end of next week, this will look much better because we'll get more images and we can average them. But yeah, all right. So I think with that, I'll end there. Say thanks for coming. I think we still have a lot of beer, so and you guys are welcome to stay, and I'll stay here to answer any questions. I don't know if we.
Thanks.
I guess.
Yeah, maybe we'll please stick around.
Mhm.
Ask Sergey more questions. We'll kind of avoid questions from the audience at the end. We got a lot during. I'll just say that we're back across the Charles next month. And Aaron Yang is going to be our speaker. So another recent Boston transplant from the IPD. And so we'll be back on the Harvard Medical School campus. So yeah, thank you there.
Awesome. Thank you, guys. Right. Oh, I guess I never came back to this thing, but all right, wait, just in case we have conservation and couplings for MPNN. So if you guys are curious, you can come back to that later.
That's interesting because we're actually looking at this ourselves too, because so if you take ProteinMPNN sequences and then just depending on the noise level and the temperature, and then compute a pause model on the sequences. You can recover decent coupling if you do the same computation on the MSA. And but not always. And not every strong coupling is recoverable. So it's quite interesting. We'll talk about it. We'll talk about it another time.
Yeah, so in this case, it is indeed, so this is not you sampling sequences and computing. This is directly just extracting it from the model itself.
I want to
Yeah, so
challenge
you can try to compare that.
You have
Yes. Okay.
you have a line of people. So I'm not
I wonder if we should. All right, thanks you guys online. I guess we'll chat in the future.
Thanks, Sergey.
Thank you, Sergey. That was great. Thanks.